All right, well, good afternoon. I'm going to introduce you to NuSirt. There we go. We are a small company with a vision towards preventing and treating age-related diseases, like most folks up here. We do so, however, using well-characterized compounds with established safety profiles, and we do so by optimizing energy sensing, which is something you've heard a lot about already. Our primary focus is cardiometabolic disease, and we have conducted, or successfully completed, I should say, 3 phase 2 studies with therapeutic combinations in obesity, NASH, and diabetes. We also have on the consumer product side 4 phase 2 equivalent studies using natural product combinations, as well as a fairly deep preclinical pipeline. You've all seen quite a bit about energy sensing, so I don't really need to spend much time on this slide. Safe to say that our focus, you've already heard about mTOR just on this panel and earlier during this meeting. We're going to be focusing on AMPK and SIRT1, and in the interest of time, I'll skip past, and we're going to do so on a platform built around the amino acid leucine. One of the things that we have shown is that leucine is a direct activator of leucine, which in and of activator of SIRT1, which in and of itself is not sufficient to generate therapeutic effects, but which instead enables robust synergy with SIRT1 activators, and I should say SIRT1 pathway activators, both direct and indirect activators, as well as with NAD donors. This is a unique feature of leucine. The other branched chain amino acids do not have this feature, nor do any of the non-branched chain amino acids. But leucine-SIRT1 combinations very nicely mimic the effects of mild caloric restriction, and although there's a substantial literature on leucine and mTOR and TORC1, choosing the right concentrations enables you to accomplish this without any activation of mTOR. And so we have a platform that is built around leucine combined with a series of co-activating compounds that the co-activating compounds act either directly on SIRT1, on AMPK, or on eNOS, or a combination thereof, and in doing so allows pleiotropic effects on a range of cardiometabolic diseases. We think about this as a continuum of solutions. If you think about solutions for age-related diseases, you can go from the left side of what we would call unsupported noise in the nutraceutical marketplace to trusted and validated brands, which is one of the things that we seek to do, which is natural products with solid clinical trial data to address age-related diseases, all the way to the right-hand side of the slide to true therapeutic solutions, that is RX. And our intellectual property covers the continuum from trusted and validated consumer products through therapeutic products. So just to give you a top-line view, on the natural product application, we have 2 that have been through clinical trials. That one is a combination of leucine and resveratrol, and the other is a combination of leucine and pyridoxine, vitamin B6. The preclinical data show really strong improvements in insulin sensitivity, as well as reduction in inflammatory biomarkers. We see very nice reductions in weight, visceral adipose tissue, total body fat, and in preclinical models, of course, we see lifespan extension. On the clinical side, the leucine-resveratrol combination has worked well in increasing insulin sensitivity, increasing glycemic control in pre-diabetic subjects, while the Leucine-B6 combination has worked very well in augmenting weight loss during mild caloric restriction, enabling a 9% weight loss over a 6-month period of time, with associated improvements in insulin sensitivity and reductions in inflammatory biomarkers. Parenthetically, I'll say, if we hold weight constant and do not allow the subjects to lose weight, we instead see a shift in body composition from adipose tissue to lean mass. So with a fixed body weight, there's a reduction in adipose tissue and an increase in lean mass, and again associated with reduction in inflammatory biomarkers. Now, for therapeutic applications, we've gone through actually 3 different phase 2 programs. Right now, our lead is a triple combination of leucine, and the doses are shown here, 1.1 gram twice a day, metformin at 500 milligrams twice a day, and an extremely low dose of sildenafil, 1 milligram per day. We think about sildenafil more along the lines of 25 to 100 milligrams of sildenafil in the form of Viagra as a PDE5 inhibitor. At this much lower dose, you're really focusing on its activity to activate eNOS. Preclinically, we've shown that this triple combination increases SIRT1 and AMPK signaling quite a bit, increases fatty acid oxidation, reduces both adipose tissue and liver fat accumulation, and does so much more than any of the dual combinations or individual components. Clinically, we see a number of things, and I've tried to consolidate 3 phase 2 trials into this single slide. What you see on the upper left is in a study of 267 patients. The purple-colored line is our best dose of Leu-Met-Sil, and we see nice weight loss in the absence of any caloric restriction, lifestyle change, or lifestyle advice. That was a little over a 2% to about a 3% weight loss. One of the things that we noted in that trial was that there was a subgroup, that is African American subjects, and we enriched in African Americans, had twice that level of weight loss. And when we went back and asked why, we landed on insulin. Once we controlled for insulin, or really insulin sensitivity, as you can see on the upper right, we get a much more robust loss of weight in individuals with high insulin, modestly high insulin, or high HOMA, and here we're just using insulin as a surrogate for HOMA. So we get very nice weight loss, especially in insulin-resistant individuals, and that is regardless of race. In the lower left, you will see that in subjects with non-alcoholic fatty liver disease, we get about a 15.5% drop in liver fat. And although I couldn't fit it on the slide, that is also accompanied by a regression of the lipidomic signature for NASH. So it appears to be a good and viable candidate for NASH. But we've also seen a number of other cardiometabolic benefits from this triple combination. 5.5 millimeters drop in blood pressure overall, but 8 millimeter drop in hypertensives. 40 milligram per deciliter drop in triglyceride, but about 3 times that drop in those with elevated triglycerides, and about a 50% improvement in insulin resistance as measured by HOMA. So the red light is blinking, so I'll read my summary for you. So basically, we have a platform that shows that leucine activation of SIRT1 results in synergy both on the natural product side as well as the pharmaceutical side to address cardiometabolic diseases associated with both age and Western lifestyle. We've got clinically meaningful results in obesity, NASH, and cardiometabolic disease. We have, and this is important, a demonstrated positive safety profile because we are using combinations of well-understood components with well-established safety profiles to enable both our consumer health and therapeutic applications. And finally, the last bullet, these combinations are all protected thoroughly with a series of patents covering both compositions of matter and methods of use. Thank you.
Thank you, Michael.